Hello, my name is Dr. Marian Olpinski, MD, PhD, a pediatrician from Chicago suburbs. Today I want to talk to you about a very promising new vaccine, ChAdOx1nCoV-19 from Oxford University in England, trying to predict if this vaccine may be the first approved against COVID-19. So, how vaccines work? The purpose of vaccines is to create immunity against microbes, such as viruses or bacteria, without causing disease, which those microbes would normally cause. Vaccines contain proteins or proteins combined with sugars, so-called glycoproteins, which are called antigens and which, when introduced into the human body, mainly by injection, create immune response. Humoral means producing antibodies, or cellular, involving lymphocytes T, which are not shown on this slide. And when the real microbe attacks our body, those antibodies are already ready to defend us. Since 1796, when a British doctor, Edward Jenner, noticed how milkmaids caught mild cowpox but rarely went on to contract the deadly smallpox. Until now, all the vaccines were made the same way, by introducing antigens into the human body. Before I explain how the new vaccine will be made, let me talk for a while about SARS-CoV-19 virus causing the present pandemic of COVID-19. The virus carries his genetic code written in ribonucleic acid, wrapped in proteins and lipids. Sticking outside the wrapping, here in red, you can see a very important protein S, shown here on this picture, with its crown shape, in Latin corona, which gives this group of viruses its name, coronaviruses. Another name of that protein is a fusion protein. It allows the virus to fuse with a very specific receptor on human cells, which is called ACE2 receptor. ACE2 receptors are present in many human organs, but the virus enters our body via respiratory tract, through the mouth, nose, or eyes. The fusion with ACE2 receptor is the only way for the virus to infect our cells. If we could block the fusion, the virus will be harmless. So, once again, if we could block those spike proteins here with the antibodies, this virus couldn't attach to this receptor, couldn't fuse to this receptor, and it couldn't go inside and starts to replicate itself, building the new particles of the virus, and starts attacking our body further. And its code is written in this part of the viral RNA. It contains about 3,800 letters, and it codes this structural protein, spike protein. More than 100 companies are trying to develop a vaccine against COVID-19. Most of them are trying to do it using the conservative methods, introducing viral antigens into human body. Only few have chosen a different path. So what different path I'm talking about? For the last 30 years, 30 plus years even, scientists were thinking about nucleic acid vaccines. Instead of delivering viral or bacterial proteins, antigens, into the human body, they wanted to deliver information about the build of the protein, antigen. The information contained in our genetic code in nucleic acids. The problem was they didn't know how to do that. It's not an easy task. Scientific progress in the last few years has changed it. Now we know how to do that. There are few groups working now on nucleic acids vaccines. Two of them, American company Moderna and a group from University of Oxford, sorry, it's written in Polish, I have this lecture in Polish too, have already started their COVID-19 vaccines in clinical trials. To deliver the genetic code of the protein, in our case, part of SARS-CoV RNA coding spike protein S, into the human cell, those two groups use two different vectors. Vectors means packages for the S protein gene. Moderna packed this piece of RNA into a lipid nanoparticle, and Oxford group uses a manipulated and harmless animal virus, inserting into it the gene of the spike protein. This is the manipulated virus, vector virus, which I will be talking about in a few moments. So, what virus it is? You can see here a paper published in 2012 by a group led by Professor Sarah Gilbert. They found a chimpanzee adenovirus Y25, which causes common cold symptoms in chimpanzees. Using a very sophisticated genetic engineering methods, they changed the virus so it can't multiply, but can be used as a messenger vector to deliver genetic information about given protein into a human cells. What is very important, the virus which we want to use as such vector should not be neutralized by our human immune system. It has to be foreign, but harmless to us. We shouldn't have antibodies against it. And it was proven in UK and Gambia. After the biomolecular modifications, this chimpanzee adenovirus Y25 was changed into ChAdOx1, chimpanzee adenovirus Oxford 1 virus. This is the name, which is harmless because it cannot replicate, so it cannot cause infection in a vaccinated individual. It is a very well tested as a viral vector and has been safely used by thousands of people from 1 week to 90 years of age in vaccine trials at various stages against over 10 different diseases, including MERS coronavirus, Congolese hemorrhagic fever, Zika virus, Lassa virus. So how it works? Because ChAdOx1, this is the ChAdOx1 virus, is a DNA virus. It means its genetic code is written in DNA. The genetic code for S protein, so this is a S protein from the SARS-CoV-2 virus, and this code for the virus is written in this part of SARS-CoV-2 RNA. This frame shows it. So this code for the spike protein has to be first reversely transcribed into DNA, but it contains the same information about this protein. This piece of DNA is then inserted here into the DNA of ChAdOx1 vector, and the vaccine is made. It is then injected into the arm, muscle, and delivered into human cells. They will start making spike proteins of SARS-CoV-2, which, as foreign for the human body, will trigger immune response, humoral production of antibodies, and cellular production of lymphocytes T, which will remember the shape of this protein and will be ready to fight when the real virus attacks us. So how it is possible that Oxford group is already in phase 1 and 2 of clinical trials? How it is possible that it took them so little time? The answer is very simple, but before I will give you this answer, once again this schematic picture how the vaccine is made. Once again, spike protein of the virus, the information, the genes coding the spike protein are cut from RNA. There is a gene sequencing, and this gene is packed into the modified Chimpanzee Y25 adenovirus, which name is changed into this, and then the vaccine is made. The vaccine is injected, and then the cells start to express spike protein. The body starts producing antibodies, and this person should be immune. So when the real virus wants to attack, this person will not get sick. It's so simple. So how it's possible that they are so ahead? Because they already tried this type of vaccine against very similar virus, MERS coronavirus. Remember, Middle East respiratory syndrome caused by MERS coronavirus which is quite similar to SARS-CoV-2. In 2012 and in 2013 caused a very deadly disease which affected around 2,500 people, killing 35% of them. This virus is still endemic in Middle East. This paper describing the trial on 21st volunteers who received this vaccine was published on April 20th this year. It was published 12 days ago. And what is the result of this trial? So you see that they used the same vector, ChAdOx1, but they inserted the genetic code of MERS coronavirus spike protein, and after a single dose, this vaccine was able to elicit both humoral and cellular responses against MERS coronavirus. The result of this first in-human clinical trial support clinical development of progression into field phase 1b and 2 trials. So what happened now? The group of Dr. Gilbert, you can see her name here again, just inserted, they replaced the code for S protein of MERS coronavirus with the code of S protein of SARS-CoV-2. This code was first known on January 10th, 2020. In March 2020, 6 monkeys, 6 rhesus macaques, were given ChAdOx1nCoV-19 vaccine, and then after 28 days of heavy exposure to the virus, none of them got sick. It was proven earlier, I have to emphasize, on another group of unvaccinated macaques that COVID-19 virus can make them sick. After that result, that achievement, a first human trial of vaccine began in UK on April 23rd, 2020. So this study will be determining efficacy, safety, and immunogenicity of this candidate coronavirus vaccine, ChAdOx1nCoV-19, in UK healthy adult volunteers. Those volunteers will be between 18 and 55 years, and total 1,112 volunteers will be enrolled, and those volunteers will participate in the study for approximately 6 months, with the possibility of prolonging it to almost 1 year. So how it will be done? So half of the group will be given 50 billion viral particles of ChAdOx1nCoV-19, the vaccine, which is being tested, and the other half will be given a known vaccine against meningococcus, the bacteria which can cause meningitis, and this name of vaccine is ACWY, which is a known vaccine given for the last 5 years, at least in United Kingdom, So why the scientists do that? So people who are getting the vaccines, of course, they don't know which vaccines they get. And we give the real tested vaccine to around 500 people, and around 500 will get this meningococcal vaccine. The scientist wants to compare so-called side effects. We know that vaccines can give very mild side effects like pain in the arm, little fever, muscle aches. So they want to compare if there is a difference between those side effects. That's why half of the people are getting this known meningococcal vaccine. There will be also one group which will be getting a booster after the first dose. And there are, of course, many other criteria which we don't need to talk about now. So how we will know if this vaccine works. So in the next 2 to 6 months, we will know, because those people will go to normal life. And some of them will get infected with the COVID-19. And between 2 and 6 months after they are injected with the vaccines, they will be checked. They will be checked with the test, the viral test, the PCR test, and we will know how many of those who were vaccinated with ChAdOx1nCoV-19 vaccine get sick, comparing with the group which got the meningococcal vaccine, which of course can't prevent people from getting COVID-19. There will be also, those people will be checked for serious adverse events, because if those serious adverse events occur, this trial will have to be stopped. And why I'm talking about 2 or 6 months? It only depends how fast is the spread of the virus in the community. If the spread is still high, if the spread is still very prominent, we will get the results faster. And if the spread will be much slower, then we will have to wait for the results longer. I think that we will not need to wait so long, because the spread in UK is still quite high. So here I can show you Elisa Granato, the first volunteer being injected with ChAdOx1nCoV-19 vaccine. And now I want to answer the question I made at the beginning. So for me, it's a very promising vaccine, and in my opinion, it has a very good chance to be successful and maybe even to be first approved vaccine against COVID-19. We really need it. Thank you so much.